What if I told you that a simple pea-plant experiment holds the key to nailing 3, maybe even 4 direct questions on your CUET biology exam? So many students get completely tangled up in all the ratios and end up losing really easy marks. But I promise you, stick with me to the end of this explainer and you will absolutely master the code of Mendelian genetics. Let's get right into it.
Okay, so why are we focusing so hard on this topic? Here's the deal. Mendelian genetics is the absolute foundation for your entire biology syllabus. The CUET exam setters, they love to test you on monohybrid cross ratios, the crucial difference between phenotype and genotype, and the exact definitions of Mendel's laws. Mastering this isn't just about grabbing a few marks here, it's about building a solid base for all the advanced genetics that comes later.
So, what is the big trap here? Where does everyone get stuck? Honestly, it all boils down to one simple point of confusion. The difference between what you can physically see in an organism versus what's actually written in its hidden genetic code. This is the exact hurdle where most students stumble, and today we're going to clear it once and for all.
Look at this slide. This is the heart of the mystery. On one side, you have the phenotypic ratio, 3 tall plants to 1 dwarf plant. This is what you can see with your eyes, right? The observable outcome. But on the other side, we have the genotypic ratio, 1 to 2 to 1. This reveals the secret, the underlying combination of genes. Understanding how we get both of these from the same experiment, well, that's the key. And it all started with one man and his garden. So let's meet the detective in our story. Now, this guy didn't have any fancy labs, no DNA sequencers, no supercomputers. What he had was a garden. A really sharp, methodical mind, and the absolute perfect subject for his investigation. The common pea plant.
And here he is, Gregor Mendel, the one and only father of modern genetics. Through thousands and thousands of meticulous experiments on these pea plants, Pisum sativum is their scientific name, he single-handedly figured out the fundamental laws of heredity that we still use today. He's the one who cracked the code.
But why pea plants? I mean, why not something else? Well, this was a stroke of genius. They're easy to grow, they have a short life cycle, which means you can see results across generations really quickly. And most importantly, they have very clear either-or contrasting traits. Think tall or dwarf, round seeds or wrinkled seeds. There were no confusing in-betweens. This gave Mendel clean, quantifiable data to work with.
All right, let's get into the nitty-gritty. We're going to break down the central experiment that unlocked everything, the Monohybrid Cross. Listen up, because every single step here is a potential exam question. Pay close attention.
So what in the world is a monohybrid cross? The name itself gives you a huge clue. Mono means one, right? So it's a cross where we're only tracking the inheritance of one single trait, like plant height. By keeping it super simple, Mendel could isolate the rules for how that one specific trait was passed down. So here's how it plays out, kind of like a 3-act play. Act 1, Mendel starts with 2 purebreds, a tall plant with the genes TT and a dwarf plant with genes TT. We call this the parental or P generation. Act 2, he crosses them, and guess what? Their offspring, the F1 generation, are all tall. The dwarf trait just seems to have vanished. Poof, gone. But then, for Act 3, he lets these tall F1 plants self-pollinate. And in their offspring, the F2 generation, bam! The dwarf trait suddenly reappears. So, how did that happen? And this beautiful little box explains exactly how. This Punnett square is our decoder ring. So we take the 2 possible genes from one parent, a big T and a little t, and we put them across the top. We do the same thing for the other parent down the side. Now it's just like a simple multiplication table. Top left, big T meets big T, you get TT. Top right, big T meets little t, you get TT. Bottom left, little t meets big T, we always write the capital first, so TT again. And bottom right, little t meets little t for TT. And there you have it, that's the genetic result.
Okay, now let's connect this back to what Mendel actually saw in his garden. Because the allele for tallness, the capital T, is dominant, any plant that has at least one capital T is going to be tall. So the TT plant is tall, and both of the TT plants are tall too. That's 3 out of the 4 boxes, right? 75%. Only that one little TT plant is dwarf. That's 1 out of 4, or 25%. So the ratio of what you can see, the phenotype, is 3 tall to 1 dwarf.
But, and this is a huge but for your exam, here is the hidden code. Let's look at the actual gene combinations, the genotype. We have one TT, that's homozygous dominant. We have two TTs, those are heterozygous. And we have one TT, that's homozygous recessive. So this gives us a genotypic ratio of 1 to 2 to 1. Please remember this. The CUET exam loves to ask for the genotypic ratio, and so many students just automatically write 3 to 1. Don't let that be you.
Now, what's so amazing is that from this one simple experiment, Mendel figured out two of the most powerful and fundamental laws in all of biology. These are the rules that govern heredity. The first one is the law of dominance. This is the reason why all those F1 plants were tall, even though their genetic code was actually TT. The dominant allele, that capital T, completely masks the effect of the recessive allele, the little t. The dwarf trait was still there in the genes the whole time, it was just hiding.
Okay, let's pause here because this concept is absolutely non-negotiable for the exam. Phenotype is the physical appearance, what you see. Genotype is the actual set of genes, the hidden code. So this means a tall plant can have a genotype of either TT or TT. Getting this distinction right is everything.
His second major discovery is the law of segregation. This explains how that dwarf trait suddenly reappeared in the F2 generation. What it says is that an organism's two alleles for a trait, like big T and little t, don't blend together like paint. Instead, they separate or segregate when sex cells, gametes, are being formed. Each gamete ends up with only one of the two alleles. And because those alleles separate so cleanly, this law is also called the law of purity of gametes. And honestly, that's a great way to remember it. It just means each gamete is pure. It's only carrying one version of the gene, either the big T or the little t, but never both. So here's the crucial point. If you have a tall plant just sitting in front of you, you can't tell by looking whether its genotype is TT or TT. So how could Mendel actually prove his theory was right? How could he confirm this hidden code he was talking about? This was the exact challenge he was up against. He needed some kind of experiment, a way to reveal the hidden genotype of an organism that was showing a dominant trait. And once again, he came up with a simple and totally elegant solution.
He called it the test cross, and it is a brilliant method. You take your mystery plant, the tall one you don't know the genes of, and you cross it with a homozygous recessive plant. In our case, that's a dwarf plant, genotype TT. The results of this cross will tell you the secret. If all the offspring come out tall, your mystery plant had to be TT. But if you get a mix of tall and dwarf offspring in a 1-to-1 ratio, you know for sure your mystery plant was heterozygous, TT.
All right, it's time to put your knowledge to the test. I'm going to give you a CUET-style question. I'll read it out, then I want you to pause and try to answer it yourself. Ready? Okay. Question 1. The genotypic ratio in the F2 generation of a monohybrid cross is what? Is it A, 3 to 1, B, 9 to 3 to 3 to 1, C, 1 to 2 to 1, or D, 1 to 1? Pause now and think. Okay, the correct answer is C, 1 to 2 to 1. Remember, 3 to 1 is the phenotypic ratio. Do not get those mixed up. Great job. Okay, let's quickly talk about the common traps students fall into. First, confusing genotype and phenotype. We've hammered this home. Genotype is the genes, phenotype is the physical look. Second, writing 3-1 for the genotypic ratio. No, we know it's 1 to 2 to 1. Third, thinking dominant means stronger or better. It doesn't, it just means it masks the recessive allele. Avoid these and you are in fantastic shape. And now for your quick revision. Here's your toolkit. Burn this into your memory. A monohybrid cross tracks one trait. Phenotypic ratio is 3 to 1. Genotypic ratio is 1 to 2 to 1. Alleles separate because of segregation, and one allele masks another because of dominance. You master these 5 points, you've mastered the basics.
Okay, challenge time. Here's a bonus question, a highly expected one for the CUET. If a heterozygous tall plant, that's TT, is self-crossed, what proportion of the offspring will be homozygous tall, that's TT? Pause and picture that Punnett square. The answer is 1/4. Only one of those 4 boxes is TT. It's amazing to think that these simple rules, which Mendel discovered in his garden, are the very foundation for understanding all of genetics, from a single pea plant to the incredible, complex diversity of all life on Earth. If you are ready to become a genetics expert for the CUET, make sure you're subscribed. In our next explainer, we're going to tackle the dihybrid cross. I'll see you there.